I'm going to talk about mathematical immunology, which is not so common. It's, as the name make you guess, a branch of the immunology devoted to formal approaches, mostly mathematical, so computational. I will talk about my work on the topic, but I will try to give you an idea of what are the challenges in this domain, which is quite recent, I have to say. So, well, maybe you will have ideas about mathematical works you can do on this topic. So this is a bit of history. Mathematical immunology, it's a young field with respect to immunology, also with respect to mathematics, of course. Here, if you see it, I've made research on the two words, mathematical immunology. You always find all occurrences of these words. If you look at the papers, well, it's very general. Mostly work are in the early 2000s. And why at this moment? Well, immunology by itself, it's a part of biology which is quite recent too. A lot of the main discoveries in the field. The y-axis, it's the number of paper in the request. And the main discoveries date back to the 80s, okay, where we identified most of the things we know now, and it's still a very dynamic field where each year we discover new things about the immune system. So a few years back, I would have been pretty sure that almost no one knew anything about immunology in the room. Well, then there was the COVID pandemic, so probably you know something about immunology. Anyway, what happened is that mostly at the beginning of the, say in 2001, maybe the end of the 90s, people in mathematics or physics realized that in immunology, you could have access to very interesting information in the form of, let's say, temporal dynamics, cell count evolution over time, and that the underlying systems in immunology were highly controlled systems. So you could have, well, the idea of dynamical system and nonlinear problems, controlling the system, understanding how they work. So then it started around this time. And it's been mostly dependent on the nature of the data available in this field. And this is my, let's say, my summary of it. But up until, let's say, 2013, more data in the field, there were blood cell counts, measurements of concentrations of different molecules, like very basic biological information. And then around 2014, everything changed because new techniques appeared. So they're called single cell RNA sequencing. And these techniques changed the way you can look at cells. And well, I think it's a reshaping of modern biology. It corresponds to an increase in the amount of paper mentioning mathematical immunology. Why? Well, because these techniques require a lot of small tools and then increase of paper. So basically, what is an immune response? It's a response that is triggered when the organism detects that there is a threat. Okay, so the threat can be a virus, for instance, but not only virus, bacteria, whatever. And so there are cells in the organism that detect the threat, and these cells, let's say macrophages or dendritic cells, cells of the white cells, maybe that talks to you. These cells, they activate specific cells. So that's what is represented on this curve, which is the kind of curve that you can learn in high school. It was not yesterday for most of us, but so what happens? You have specific T cells like lymphocytes, and these cells are going to be activated by the cells that found the threat. And what activated means? It means that these cells are going to proliferate a lot. So here what you have is the temporal scale, and then a number of cells, but it's a virtual one. Okay. And so after activation, the number of specific immune cells will increase. And it's not only an increase in numbers, it's also a differentiation process. That is that cells will change. Okay. First, they are called naive cells, and then they will acquire specific attributes that will allow them to eliminate the threat. How? Production of antibodies, for instance, or another cell. So that's how the immune system eliminates the threats. And so you reach well, okay, there is a plateau. You can imagine that there is a maximum number of cells, and then at some point, the total number of cells shrinks. Most cells will die. If they have been produced as a reaction to a threat, and then they will die. Why? Because they are also a threat to the organism. You know, if you have cells that are able to kill other cells in your organism, it can be bad for you. First, it's good, and then it can be bad. And in the end, meaning 3 weeks, 4 weeks later, the organism should have produced new cells that are called memory cells that will remember the infection and will be able to target the threat next time you encounter the threat, okay, like the COVID virus. Okay, this is a very simple representation of how the immune system works. It's simple because it looks simple first. You know, you have cells, you activate them, they do their job, then in the end it works, and you have an immune memory. And what this kind of scheme doesn't show is the complex mechanism that can lead to the production of this very specific and kind of dangerous population, and then control it. So maybe we can say that, I can say that not all organisms on Earth have a specific immune response, for instance. It's not necessary. We have non-specific actors of the immune response, like the skin, which is the first barrier, and then different things that are absolutely non-specific. But the specific response, it helps preventing infections very quickly. Yeah, but it's also a bit dangerous. Okay, so apart that our survival depends on that. Why is it important? Well, because if we can control it, of course, we can provide cures and we can improve the human health, for instance. But how does it work? Well, if we want to understand this, then we need to understand what are the differentiation pathways. What does it mean? It means that when cells are activated, some things happen to them. Okay? And we need to understand what happens and what will be the result. Because if we understand it, maybe we can control it. And well, maybe you have absolutely no idea if this is complicated or not, but it may appear simple at first, and actually it's not. And it's been one of the main questions in immunology over the past 20 years. How can we define correctly the differentiation process and then have well-defined subpopulations on which we can act? If we can do that, then one of the most important questions is the second one. Is it possible to predict a long-term response from early molecular events? This is about vaccine design, because when you develop a vaccine, what you create actually is something that will stimulate your immune system, but at a molecular scale. You will activate cells. That means that some genes will be activated and will result in something. But what you want to observe is something that occurs weeks later. It's this entire differentiation process. Okay. Hopefully, what you want to obtain is an important memory cell population, meaning that you will be protected against the infectious agents. So this is a very complicated question that I will address in part. And the last one, of course, is how can we account for inter-individual variability in the immune response? Because we could say this is a classical question when you work on living systems. You know that most of the time the variability is important between individuals, inside individuals, whatever. And most of the time, that's not the first thing we focus on because it's too difficult. And so we consider average behaviors. But then in the immune system, you can have nice drawings like that and think that this happens in every individual, always like that, with the same temporal scale, the same physical scale, whatever. It's not the case. The variability is very high in humans, but not only in humans. I think I will show you data on animals, which are supposed to be all the same, and the variability is very high, actually. So these are the three main questions. And why did people use formal tools to try to answer these questions? I think it's because it's very difficult to access the immune system in its entirety and over long time scales. Because I didn't say that, but let's imagine you get the flu. How does it work? The virus goes into your lungs, it infects cells, it kills cells, it spreads. And after killing many cells, then it's detected by, let's say, the surrounding cells that are able to identify them as a threat. And then these cells will circulate in the body, go to lymphoid organs, like the lymph nodes that we have almost everywhere, and try to activate cells. And then these cells, they will proliferate, and at some point they will leave the organs and circulate again and find the site of the infection, and then they're supposed to act efficiently against the infection. And so this takes time, this takes a lot of energy for the cells, and it may not work for different reasons. I said that this is a kind of dangerous process, the immune response, because you produce cells that can target your own body. So there are a lot of control mechanisms. For instance, if the stimulation is too strong, it may mean that actually there is no threat, because if it's too strong, it means that it's easy to detect. And if it's easy to detect, maybe it's yourself. Yeah? So you don't want the system to react too quickly. No, you don't want the system to react if it's highly stimulated, and you don't want it to react if it's very weakly stimulated either. Okay, otherwise you will react all the time. It's easy to say, it's not so easy to understand how it works and to identify the specific mechanisms. Okay, so I will first, but most of all, discuss this part, which is about how to identify the differentiation pathways in a specific immune response based on the CD8 T cell response. And I hope it will be a lot about modeling, but I hope it will give you hints about what are the limitations, what we can do, what we can do. Very brief state of the art. The first work on the modeling of the specific immune response. So it dates back to 2001. Rob de Boer was a colleague from the Netherlands. And so basically the question was, if we measure in, here it is in mice, if we measure the number of lymphocyte T cells over time, so this is a log scale, we have few measurements, and we see the kind of dynamics that was represented on the previous slide. So the number increases, then decreases, and it stabilizes higher than it was at the beginning. Well, can we characterize these dynamics? That was the question. You know? Basically, we say cells are activated, they differentiate, and can it work? And so they proposed a very simple model. It's a bit funny, it's written like that, but actually it's a very simple system of ordinary differential equations, because it just says that here, then cells are activated, and then cells die or differentiate. So it may look a bit complex, but actually it's not. So this is a system that you can solve. Okay, so you can make students work on it. And they managed to feed the data, and they identified parameters that were very important. And so their parameters were the time at which the stimulation starts, these kind of things. Unfortunately, the same year, biological evidence showed that it doesn't work like that. So the mechanism they implemented in the model, well, they're not okay, basically. But okay, this information was not available. They proposed something. So then from this work, I will mention only one work, but a lot of people try to improve this and account for the existing knowledge and try to be more accurate in their description of the immune response dynamics. Well, so far it works. Well, I mentioned this work because I think it's an interesting first work where people try to use a partial differential equation in the modeling of the differentiation process. And so they said, okay, it doesn't work as modeled by the previous authors, meaning there's not activation point and an end time. It's something that is more or less continuous, even if you activate it, but just for a brief moment, you can trigger an immune response. So it's a bit more complicated. And so they try to use partial differential equation to account for the fact that the activation may take some time. And so there is a structure in the population of cells that is supposed to account for that. The main issue at the time was that, and it's still an issue, it's very tempting to use partial differential equation to model a differentiation process, because you can see differentiation as a continuous process. And since you're focusing on a cell population that will evolve in time, you would like to have both a time structure and, let's say, a differentiation structure, something that is associated to differentiation. In modern biology, differentiation is associated to the expression of genes. So you could say it's a gene marker that you will measure over time in the cell population. You could say differentiation is a discrete process, but that will be an old view of differentiation. It is, in fact, discrete because we measure only discrete events, but there are a lot of indications that actually it's a continuous process in the expression of genes. And so partial differential equations, I think they are a good tool to describe this process. However, to validate the model, you need data that are not available. We don't have distributions of the cell populations according to markers, at least not very finely defined. We have something that is very raw, so it's a bit complicated to use them. I won't talk about recent works on that, but this is something that I've been interested for years, trying to have a model based on partial differential equation that would account for the differentiation of CD8 of lymphocyte cells. And it's possible to write models, it's possible to study them, it's very difficult to validate them. This is not specific to the immune response. Okay. And mostly the models became more complex, but the data remained the same. So you still wanted to fit 5 points. Okay. With more and more complex tools. And then data changed, and most people stopped working on this kind of approaches. And I will go back to that later. So first, just a word, if you want to, what is a biomarker? Because I will talk about that. So I told you how differentiation is defined. Well, if you are able to take a cell out of an organism and look at this cell, well, you will look at the membrane of the cell, okay? You will, let's say you will not look inside the cell. So at their surface, cells express proteins. Biomarker will be a specific protein that you are able to identify on the surface of the cell. And this is how cell populations are defined. You will say, this is the population of cells expressing the red and blue biomarker, or the green one, or whatever, except they have not very funny names. Okay, but that's how it works. And that's what I'm going to talk from now on. So the questions that has been the focus of almost the entire community in immunology is how can I identify the biomarker characterize this differentiation pathway when cells are initially in a naive state. Naive means they have not seen any antigen before, any infectious agent, and they're just waiting to be stimulated. And memory cells, these ones are the results of the entire immune response. So these are the cells that will remember the infectious agents and that will be able to act efficiently against a new infection. So once again, why do we want to control that? Well, because we want to activate naive cells and generate memory cells, and we want to do it efficiently. So it's probably necessary to understand how it works. So this means, can we identify biomarkers that will characterize these two states and the states in between? You know, the differentiation states that are necessary to go from naive cells to memory cells. So we'll go briefly on that because it's biology. I'm not sure it's what you're interested in, but in 2012, researchers in the United States identified two biomarkers that are called CD127 and KLRG1. And they said that by focusing only on these two biomarkers, so it means two kind of proteins expressed at the surface of cells, you could identify the populations of interest in the immune response. Because you can identify what they call early effector cells. Effector in immunology, it means that the cells will be able to act against infected cells. You remember the drawing at the beginning? These are the cells that are most represented at the peak of the response. And so they said, with these two markers, you can identify this population, and then you can identify the ones that are going to kill the infected cells and the ones to generate a memory population. Except it's not that great because actually this population is quite well defined, but this one, so the name is memory precursor effector cells. The word which is important here is precursor, because it means that these cells are not exactly memory cells, but they should become memory cells, provided that, well, they survive, they differentiate again, whatever. They're still not exactly memory cells. Yeah. And so actually, it's a very heterogeneous population, which does not clearly characterize a memory cell population. Consequently, it's not a very efficient target. Well, and then from this characterization of the differentiation process, many people suggested other pathways. And then, to summarize the debate, it's been about, is the differentiation process a linear process or a branching process? Meaning, are naive cells differentiating one step at a time in different kinds of cells and in the end generate population, or is there branching initially? Some cells will become memory cells, other cells are doomed to be something else and die. It may seem a bit naive, in the view of what is the differentiation process. I remember my opinion was we don't really care whether it's linear or branching. What matters is can we identify the differentiation process that is correct. But actually, it's very important for immunologists. And they thought a lot about that. So they identified, so here you have these two markers. Then people said, actually, memory cells come before the others, but they use different biomarkers. So, if you imagine that you represent your cells in a 2 or 3D space, it's just not the same space. Okay? So maybe it doesn't mean that it's exclusive. These ones, I didn't indicate the biomarkers, but they're again different. So each time you have a characterization that is different and that structures differently the entire cell population. The question is there one representation that is more accurate than the other? And based on experimental measurements, it's not possible to decide. Because it's a very static view. You measure the expression of these markers at a given time point. And you say 90% of my population expresses these 2 or 3 biomarkers. And then 5 days later, it's only 20% of the population, and you may use different biomarkers. So that's why people started to use mathematical models. So that's what have been used in the 2 previous slides. Actually, to validate the differentiation process, people try to model the differentiation mathematically by using tools from dynamical populations. And so with my colleague, we try to. So basically, this is what we have when you talk about biomarkers. What you do is that, so you take your cells out of your organism, you put them in a solution where they will be in contact with antibodies, and then you're able to measure these antibodies. And so then you put them in a machine that will count the number of antibodies, let's say, and then you project the number of cells on a 2D space where each axis is one biomarker. So here, for instance, you have 2 biomarkers, KI67 and BCL2. And what you observe here, it's still in log scale, but it doesn't matter, that each dot is a cell. Yeah? So you have a representation of your cell population in a 2D space. This is a snapshot, okay? It's at a given time point. But then you can do that at several time points. And what you observe is that the cell population changes. You agree with that? Yeah? The cell population changes. First, it's on the upper quadrant over there, and then in the end, it's no longer there. So how can that be? How can this population evolve like that? What are the rules? How does it work? So, okay, flux cytometry doesn't matter. So the question is, when we observe that, do we observe a differentiation process? So here you have cell count over time. The color means are expressed at the time. Yes?
I'm sorry, can I ask something?
Yes, sure.
Are you sure that it's the same population and that these cells are making other different cells?
That's the question. I'm not sure. Okay, but the question is, can we say that it's the case? Okay. So how does it work? First, you have your population and you select all the cells that with generic biomarkers in common. So you're sure that it's immune cells. You're sure that it's lymphocytes. You're sure that it's CD8 lymphocytes, okay? So you're sure that you work on a sample that is comprised of the same kind of cells. But then they don't express exactly all the same biomarkers at the same time. And the question is, are these cells an evolution of this one or not? So you can count this type of cells over time. So when there is a minus in front of the biomarker, it means that you're on the below a threshold value, and when there is a plus above a threshold value. Okay? This is biology. And so they're all different. This population is the red curve, for instance. So you measure this population, the one that have a high level of these biomarkers and a low level of this one, over time, and you have this curve. Then you count this type of cells, and you have the red one, the green one, sorry. And then finally, you measure this one, and you have the green curve. So you have potentially different subsets. At least they are defined by the biomarkers they express. Yeah. So it's not the same cells at every time point. Okay. The measurement obliges you to kill the cells. Yeah. But they express the same biomarkers and not the others, and in the same amounts. So you can say this is a population. Yeah? This is a population, this is another population. But now the question is, does the pink population arise from the green population? And does the green comes from the red? And the red from the blue? So you have this, and you have these curves. So when you look at the curves and at this data, you can have an idea whether it's true or not. Yeah? Because here you can have the feeling that first you have the blue ones, they disappear, but the red ones appear. But as the red appears, the green appears, and then they decrease, and then it's like a chemical reaction, but it's not. Yeah. And here you can have this feeling that actually, if there is a differentiation process, it consists in moving continuously in this 2D space like that. How can you determine if this is true or not? You don't have any other experimental data. Okay, that's all we have. But what can we do from that? Okay, it's not so easy, huh? Okay, but we have ideas. Actually, if you were my biologist colleagues, you would say, sure, this is the differentiation process. Why? Because we cannot make more accurate measurements, and this is the best information we have, and it seems that it works, so it probably works. Well, we said, okay, we can try to model the differentiation by considering these different cell populations and trying all the combinations possible in terms of differentiation. What does it mean? It means, let's assume that the blue gives birth to the red, but also let's assume that the blue gives rise to the pink first, and then to the green, or even that some cells population disappear and do not differentiate anymore, etc. This is something we can do. We can write models, we can write complex models, we can write very simple models. So of course, the conclusions will depend on the hypothesis on the model. But we can do that, and it doesn't cost as much as experimental data. Okay. So that's what we did. So first, if we assume that this is a differentiation process, it's not true by default, because it only relies on the successive appearance of different subsets, but it doesn't mean that the second one comes from the first one, okay? You can have, for instance, a low-level population that arises later, and you don't see it in the experimental data. So we cannot conclude based on that this is a differentiation process. This is done every day, okay, but we shouldn't conclude that. So what we do is try to use every model possible to reproduce the data and try to find which model reproduces the data the best. So maybe I will go fast on that. But we imagine different models, like this one. This is the linear differentiation model, the very standard one, where naive cells become effector cells and then in the end produce memory cells. But this one, for instance, it's one of the 2 or 3 models I showed you a few minutes ago, where memory cells are produced from naive cells. And on the other side, effector cells are produced and memory cells don't come from the differentiation of effector cells. You can see that if this is true, instead of this one, probably you don't have to act in the same way on cells to produce memory cells. Because it probably means that you can produce memory cells without producing an efficient effector response, for instance. Because if you just want a vaccine, you don't care. You don't have a real infection, so you don't care that you eliminate correctly the infected cells. What you want is generate a cell population. But in this case, you're obliged to validate each step, let's say. Okay. Okay, so we have a list of models, and we try to select the best model. So how? We write every model in a mathematical form using the same rules. So we use ordinary differential equation, but they're nonlinear because you have a lot of controls in these models, and if you don't account for nonlinear interactions, actually you don't have the dynamics that are required for the immune response. And we try to characterize each process exactly in the same way for all the models. And at the same time, in the simplest way possible, meaning only one parameter to characterize one model, to characterize a process. What are the process? Proliferation, death, differentiation. And basically we built differentiation models. So the difference between this one and this one, for instance, is that here effector cells can also differentiate directly in memory cells and disappear because there is a differentiation parameter here that is not present in the other equations. Okay, then we have to estimate the parameters of the model that allow to have the best reproduction of the data. What we have is this kind of information, the one that I showed you. It's cell counts over time. We don't have the same measurements for all the cell populations. The blue one, for instance, it disappears very quickly. So we have only early measurements. The other one, they remain till the end of the response. So we have more measurements. So you see, you don't have the same number of time points for each subpopulation. And what we do is we perform simulation. Okay, I'm just illustrating the minimization of the least square. It's obvious for everyone. Okay. And we try to minimize the distance. And then we use a quality of fit criterion. So what are they? They're statistical indicators that will measure the quality of your fit based on how close you are of the data and by balancing with the number of parameters you use to be good. Yeah, because if you use 10 parameters to be as good as a model with 5 parameters, then we can have long discussions, but usually everyone tends to say, okay, we should take the model that has the less parameters because we don't add too much information to the model, okay? Potentially unnecessary information. So this kind of criterion, there is the Akaike information criterion or the Bayesian information criterion. It's quite easy to find the literature on these, but basically they're computed from the log likelihood of the data, and there is a weight for the number of parameters that are used. And when we do that, for each model, we obtain the number of parameters that is necessary to fit the data, and then the error. And from the, let's say, from the Akaike information criteria, we're able to compute probabilities that the model is the best one. And so based on that, we say, okay, the linear differentiation process is the best one to describe what we observed in the data. So the linear differentiation process, it's this one, where naive cells become effector cells that become memory cells, which is the one that you find in the textbooks, for instance. But actually, if you look at the results in details, well, you can say that there's not only one model that is the best one to describe the data, actually, because there are at least 5 models or 4 models here whose performances are comparable. They're very, very close, actually. And the result is this one. In order to reproduce the data, the best differentiation model is the one where cell population differentiate in a linear way, but you cannot exclude that sometimes part of the population differentiate, let's say, quickly or early in the other cell types. Okay? You can look at that in different ways. They can skip steps in the differentiation process. So if you have this 2D space, they can go from one place to the other, let's say, probably they still go in a continuous way, okay, that we're not able to observe, but they do it so quickly that it looks like a jump. Okay, once we have that, what happens? Well, we are able to describe the data, okay, that's everything we did. But more than that, we are able to access some information that can be relevant for immunologists, because if you have a model that is able to correctly reproduce your data, you can now ask questions. For instance, if I want to reproduce all the immune response that is measured experimentally, how many time points do I need? Do I need all the measurements to determine how many memory cells I will have in the end? And that's a fundamental question, because you don't want to do that. Ideally, what you would like to do is just trigger the immune response and be able to predict the final number of memory cells. Also, if you're not lost, you would like to predict the quality of the memory cell population, but this is something that is not addressed here. It's a much more complicated question because you need to define what is the quality of a memory cell population. And basically, it's their ability to respond to a new infection. But you see that you cannot determine the quality without triggering a new response. Okay, it's a bit complicated. And so what we found is that if we account for a lot of measurements, then not all, but a lot of measurements, we're able to determine the entire dynamics of the population, but this is not so interesting. But if we have only 3 measurements that are located before and just after the peak of the response, mostly all the information is contained in this time frame, and it's enough to correctly predict the evolution of the immune response. This is interesting because, well, of course, it means you don't have to make 10 or 20 measurements. You can control which measurements you need to do, and you know approximately when is the peak, let's say around day 7 or day 8 post-infection. So you can have measurements on day 5, on day 8, on day 12, and that's enough to determine the entire response. Okay, so this was the first item on what we want to do when we focus on immunology. The other would be, if you remember, well, can we predict the cellular outcome weeks later from early molecular events? So to do that, you need to build models that incorporate different scales in the description, and mainly the molecular scale or genetic scale, depends what you want to model, and what I just showed you, something that describes cell population dynamics over long time scales. Many of you probably have already worked on multi-scale models. All the time we say with multi-scale model, what is good is to lose one scale, for instance, like we're going to homogenize everything. We're going to do that. Why? Well, because in this case, we're able to do an analysis. We're able to generate mathematical results. And when the models get too complex, well, it's not so easy. So we proposed a very simple model in which we described, so this is a cell, a virtual cell, and we consider 2 genes in this cell. So these genes correspond to the biomarkers that I showed you previously and that are used to define the population. So this gene characterizes cell proliferation and this one cell death, let's say. And so we consider a very small gene regulatory networks between these 2 genes, meaning, okay, I don't know if you know something about genes, but basically in cells, we have many genes and these genes interact with each other in complex ways. And that's how a cell will function. So here we have a very simple representation of reality, which is, let's assume there are 2 genes in a cell, and that these 2 genes will summarize all the molecular information that will help a cell to take a decision. And so we describe the dynamics of these 2 genes, and we use these dynamics as, I will say, as the velocity for a transport equation, except that I will not use a transport equation, but formally it's very similar. Okay, so you imagine, I should have illustrated that, but I showed you the characterization of the cells in this 2D space, where you have one biomarker on one axis and the other one on the other axis. And I told you a cell is a dot in this 2D space. So this is exactly what we try to do here. So these are the 2 biomarkers. And we're going to say a cell, it's a couple of values, K67 and BCL2. So we can represent a cell in this 2D space. But we're not interested in what happens at this scale. We're interested in what happens to the cell population. Okay. So at the cell population, we define rules. Okay, so in this space, K67 and BCL2, cells will start to be activated, and then they will differentiate and become memory cells. Similarly to what I showed previously of that. And so what we wrote is the density of CD8 T cells by saying that the population of cells, so these are Ki67 and BCL2, so these are my 2 biomarkers. So my cell population is structured with 2 variables, which are the level of expression of each gene. And how this density will evolve? Well, it will evolve where the cell is located in the mu1, mu2 space, and what it can do when it's at this location. What it can do is, let's say, in which direction can it move? Okay, it's not a physical space, but we can project this kind of reasoning on it. So a cell that is at mu1, mu2, its dynamics will depend on the equation I showed you previously. Okay, so these 2 genes, they're in competition. They have specific dynamics. We can discuss them if you want, but let's say there are nonlinear dynamics where each gene inhibits the other one. So this is what happens where you are at the location X. And then the reaction term, let's say, it will depend on what you allow your population to do. Like, can my cell die? Can my cell proliferate? What can you do? What can, and at specific times, cells can be activated and become activated cell immune cells. So you have inputs at not only at the initial time, but you have source terms all the time. Okay, so basically this is the model. Just one word, why isn't it a transport equation? Because it really looks like a transport equation. It's simply, we didn't write it as a transport equation because we have a delay in the let's say in the velocity, in the dynamics here. And it was the time not straightforward to define the characteristics. Why? Well, because of the delay, the characteristics could cross each other. Okay, so it's not very easy. But okay, I will not say more now. What do we have with this model? Very interesting properties. Of course, the model is well posed, but also we have the existence of steady states up to 3. And what the steady states will represent? They will represent probably final states in the differentiation process. And what we obtain is a very standard result where the cell population will converge towards direct masses located at the steady states in the molecular space. So this is what we would like. We have this graph with the cells located somewhere, and we would like the cell population to go there, like the memory state, for instance. And so how does it work? It works quite well, because if you activate your cells, they will differentiate like that in this 2D space. You have the density population, and you see it move in this space, and in the end, it's in the memory phase. And this is what you would like to have, except that you lose the heterogeneity of the population. It's like a very strong selection process, actually. You may have an heterogeneous population at the beginning, but the differentiation process, it makes all your population converge toward a direct mass. So it looks nice, but it doesn't really have exactly the properties you would like. If you are close to so there is a bifurcation in this system, where you can have a Hopf bifurcation. And if you're close to the bifurcation, then what you obtain is that in the end, you obtain more heterogeneity in the population, actually, because the population oscillates around the steady state while remaining in the memory phase. And if you start again the response, it works. You're in the memory phase, you reactivate the population, so you reactivate your system, and the cell population again finishes in the memory phase, and you don't lose so much heterogeneity. But you created something, a limit cycle. Well, it's difficult to make it correspond to something experimentally. It doesn't mean that it's not like that. Okay, I will stop in 2 minutes. I told you at the beginning that in 2014, new kind of data appeared in biology. So these are called single-cell RNA sequencing. So this is a new technique where instead of measuring some biomarkers at the surface of the cell, you're able to measure the expression of genes inside cells. Actually, let's say you're able to count mRNA molecules inside cells. So how does it work? Let's say very similarly, you can extract cells, destroy the cells, and use a barcoding technique to count specific mRNA molecules inside the cells. Why is it interesting? Because you can count a lot of genes in large populations of cells. You can, for instance, measure at the same time the expression of 100 genes in as many cells as you want, almost. So this is why it's called single cell technique, because what you're looking at is really the inside of a cell. So you're able not to have an average level of expression in a population, but you have the information at the single cell level. So this has been a revolution in biology that still generates huge data sets of gene level expression, and for which you need tools to make sense of this data. Because you could imagine that, well, it's great, I can measure exactly the level of expression of a gene in a cell, so I will have a very accurate information on how this gene is expressed in this cell. And it's not what happens. What happens is that if you froze your cell population at a given time, and then you look at the expression of genes for a given gene, most cells won't express this gene. And a small part of them will have, let's say, high level of expression of this gene, meaning a lot of molecules. And if you look at another time, well, it still be the same. You will have most of the cells that will not express anything, and then only few cells that will express some genes. Why? Because the dynamics of the gene dynamics, they obey different laws. And a gene is a complex object where you have a promoter that is most of the time inactive, and sometimes it can be activated, and then it generates mRNA that is very quickly transformed in proteins. So you have a very short windows to observe actually the mRNA in cells. And so if you just froze all your cells at a given time, they are not synchronized. That's the point, they're not synchronized actually. And so you have no reason to observe actually an average value in all the cells. So our cells, they're a very heterogeneous population that are able to behave in an average way efficiently. But with this technique, a lot of questions arise. So how can you make sense of this very heterogeneous information? And basically, that's what people do. They have this data, they process them a lot to clean them, to try to structure them, and then they project them in very abstract spaces. So maybe you heard of UMAP, Unified Manifold, I don't remember. It's a projection technique. So you may have the PCA in mind, maybe, analyse en composante principale, maybe. But it's not exactly like that because it's nonlinear. And when two cells are close in this 2D space, it means that genetically they're close. That means that they have more or less the same level of expression for the genes, but you have many genes. And so the color is something that you put a posteriori on the data. So each dot is a cell, but then you project like different cell population, and it's kind of magical because then you can see structures. You say, wow, it's great. I see here, this is just an illustration, okay? I see here that the different types of cells that are ionized in some way, with this one being close to this one, and this one being closer, and not so close to this one. So it tells me something about how this cell population may interact or where they may come from. But once again, this is a very, let's say, a bit naive way of concluding. You know, you have an image and you say, wow, it works. It's not that easy because there are many steps of pre-processing analysis, and there are a lot of questions about how it works and what is exactly the information you have. So what we're trying to do is to use this kind of information, but actually, apart from the statistical parts, which are complex, the models we can develop are also a bit complex at the time. Maybe in 10 years, that we'll have something better. But this is the kind of data that now we have. Okay, I will stop and just think. My collaborators, most of the work I did there, I started it when I was in Lyon, so starting in 2007 or 2008. My main collaborators were Olivier Gandrion, who is a biologist at the ENS de Lyon, and Jacques-Louis Marvel. Jacques-Louis Marvel is an immunologist, and she's been motivated enough to work with mathematicians and to explain biology. And Olivier is a weird biologist. He decided a long time ago that formal methods were necessary in biology to understand things that were difficultly accessible experimentally. Thanks to these people, I managed to do a lot of things in immunology, and all the other people contributed in some way in all these works. Thanks for your attention.
Merci. Une question sur le système, ton système d'équation et différentiel avec le TXIJK,
Ouais.
Et que tu veux écrire une équation de transport. Ouais. Il y a le délai qui a un problème, mais il n'y a pas que celui-là. Il y a aussi que les cellules sont différenciées. Oui. Toutes les particules sont différenciées, oui. Puisque tu as une dynamique sur les coefficients.
Oui.
Et puis, non, il n'y a pas que ça. Mais on sait que écrire des équations, c'est pas aussi simple que ça.
Non, on arrive à les écrire à peu près, mais ensuite, pour s'assurer que c'est bien posé, c'est vraiment pas simple. Bon. C'était le sujet d'une thèse, et la thèse a été un peu réorientée, et on n'est pas revenu dessus, mais l'idée au départ, c'était d'essayer d'arriver à suivre toutes ces particules. Donc, il faudrait une approche un peu différente, je pense, oui. Yeah. So if we talk about humans, they first start in the lymph nodes, and most of the differentiation occurs in the lymph nodes, but it is finalized in the blood circulation, in the circulation, not only blood, in different organs. Okay, so the last stages of differentiation, they don't occur in the lymph node. My view of that is that the lymph nodes are like a privileged location for the immune cells, where they are protected. They have an environment that stimulates them and that protects them. And at some point, when they are mature enough, well, they leave because you want to act fast. and so they leave the lymph nodes. But,
So the shape will be-
oh, sure. I didn't mention it, but the main modeling difficulty when you focus on the immune system is that it's not located in one place in the organism. Actually, circulation is fundamental. Cells use the lymphatic system a lot. Maybe you checked on that, but there are some works on the circulation in the lymphatic system, but much less than in the main circulatory system. And they start in one organ, then they go to another one, and then you find them in another one. And following cells through the organism, that's very complicated. You can use dyes. You can track. You can try to track your cells in your organisms, but there are no technique that is good enough to have accurate measurements. Try to do that like more than 10 years ago. It doesn't work so well. It's a bit frustrating, actually. And so one difficulty is that because it's difficult to measure cells in organs, you measure cells in blood because it's easily accessible. But the concentration of cells in blood is much less than in the organs. So you make a lot of errors just when you measure. But choices.
Do you think that if you have 2D scans like in cancers, you have 2D like in different system, it's not possible?
Don't think so. I don't think so, mostly because cells move a lot and very fast. So it's very difficult to follow a cell that circulates, through the organism several times per minute. So it's a very complicated to follow all the cells correctly. Once they're on the infection site, for instance, then you have techniques, you can do things. You can also use imaging techniques, but they give you a snapshot at one moment, and you cannot do that. For ethics reasons, you cannot measure even in mice more than every 2 or 3 days. But everything happens in the first 3 days of the immune response. So basically, you know when it starts because you make it start. And then the next time you can measure, so many things have happened. So you have to fill the void in some way.
Pas d'autres questions. Merci encore.